Hello everybody, my name is Elisabeth Runker. During the last 25 years I was working in the Laboratoire de Biologie Cellulaire, formerly called also Laboratoire de Microscopie Électronique, as a member of the Clinique d'Ophthalmologie in Geneva. Since several years I am retired, living in Aarau, part-time working as a lecturer in the backstage of Ophtha Journal. I hope you will enjoy our session on the outer Plexiform layer. Today I would like to give some introductory remarks on some key features made over the last 50 years on research in structural biology of outer Plexiform layer. In particular, I will focus on the ribbon synapse and discuss how certain techniques have importantly contributed to connecting structure to function in the initiation of phototransduction and also in the formation of primary network in the retina. Which features render the outer plexiform layer so attractive for studies on neural networks? It is easily accessible as compared to brain tissue, and detailed insight into synaptic anatomy is now available thanks to the relatively simple circuits and the similarity of mechanism underlying synapse formation in retina and brain, especially in hippocampus. Last but not least, we have now at hand many specific markers that open a promising field of molecular approaches. This slide shows a semithin section through the frog retina, demonstrating that the anatomy of the outer plexiform layer is not readily detectable by conventional light microscopy. We just may be able to localize it close to the outer nuclear layer, bottom, and at high magnification we can barely distinguish the spherical profiles representing the synapses. The first neuronal synapses in phototransduction convert light-induced stimulation of photoreceptors, the first neurons, into electric signals and transmit them to two cell types, the bipolar and the horizontal cells, the second neurons, in the inner retina. Indeed, there exist two axes that convert signals from the photoreceptors. One axis is the vertical one, from the outer into the inner retina, involving the bipolar cells as secondary neurons. Secondly, the horizontal axis from the photoreceptors to the horizontal cells that allows the formation of an initial neuronal network by which visual signals are split into separate channels of information flow to detect objects lighter or darker than background. Thus, horizontal cells modulates the synaptic transmission from photoreceptors to bipolar cells, providing inhibitory feedback and being responsible for contrast enhancement. I think this will certainly be one of the main subjects of today's main talks. Moreover, there are additional transmission events in the OPL, where electric signals are transmitted between cone pedicles and rod spherules. This allows limited mixing of signaling between rods and cones. And then we have not to forget the functional participation of Müller cells surrounding the synapses. Now we come to our main question of today. How are the first and second neurons structurally interconnected? Abundant information is given by a look through the electron microscope. You see here two electron micrographs of an arbitrary section through the outer plexiform layer at different magnifications. A prominent axon running through the outer nuclear layer on the bottom left up to the outer plexiform layer top. This is a cone axon as deduced from its light density axoplasm and its large pedicle at the top right. Other roundish profiles at the right hand side represent rod spherules cut arbitrarily with numerous inclusions. These are interdigitations of postsynaptic cells within the photoreceptor presynaptic site. At higher magnification, we see that the cone pedicle interdigitates with cytoplasmic profiles of clear density represented by polar cell portions BP, and a darker profile with higher electron density representing a cytoplasmic portion of a horizontal cell, HC. On this micrograph, we also observe that Müller glial cells are present around the synapses surrounding the neuron cells as tiny rims to the right. The scheme of synaptic connections in the outer plexiform layer you have already seen is an almost historical one designed by Dowling and Boycott in 1966. That is, in general outline, is still valuable. Let us have now a closer look to the synapses between photoreceptor cells, the presynaptic part, and both postsynaptic cells, bipolar cells and horizontal cells. We have seen that they make close contact to each other in form of interdigitations. This is here encircled in red. These three cell types form the so-called triad. As the name suggests, three cell types participate in the synapse, namely the presynaptic terminals of the photoreceptors in yellow, the postsynaptic bipolar orange, and the horizontal cells in green. Their precise geometric arrangement contributes to the synaptic specificity. Moreover, in the presynaptic cytoplasm, we may detect an electron-dense rod-shaped differentiation called the ribbon. Ribbon synapses are specific to sensory receptor cells. Rods have one single ribbon, whereas cones may have several ribbons with various connectivities. The ribbon is an electron-dense cytoplasm structure at the presynaptic membrane. It is involved in tethering of synaptic vesicles close to the synaptic cleft between the presynaptic and the postsynaptic neurons. The ribbon itself is anchored within the so-called active zone, also called artiform density because of its shape. Both ribbon and active zones are surrounded by numerous synaptic vesicles. We come to this point later on. The various morphological differentiations within the cytoplasm and at the plasma membrane are puzzling, and their function remained vague until a new electron microscopic technique was available also for biological samples. This other electron microscopic technique is freeze fracturing. Freeze fracturing is based on physical breaking, fracturing, of frozen biological samples, including membranes. Splitting of the membrane bilayer into its two leaflets provides a planar view of their internal and external faces, as represented here on top right. Membrane macromolecules and other particles preferentially remain stuck to the cytoplasmic leaflet and not to the external leaflet. With this technique, the distribution and arrangement of membrane vesicles and particles in specific physiological states can be studied. Freeze fracturing is thus a method for studying the functional morphology of a cell. In the 1970s, Raviola and Gilula have exploited the ribbon synapse by using this technique. From their innumerable electron micrographs, they established a diagram at the right showing that besides synaptic vesicles, intramembranous particles are tethered in the region of the synaptic ridge. It also turned out that the synaptic ridge in its third dimension is a membrane fold, in fact, and its summit is the artiform density within the ribbon. Through freeze fracture studies, the precise arrangement of these presynaptic cytoplasmic differentiations has now become a functional meaning. They organize the players of the synaptic transaction, namely the synaptic vesicles and the voltage-gated calcium channels that are contained in the macromolecular particle you may see in the scheme at the right-hand side at the bottom. They organize them in such a manner that they come into close neighborhood, this means tethering together, before synaptic activity, this means exocytosis, initiated. This close neighborhood provides the structural basis for the formation of a dynamic web of molecular interactions. This is summarized in this slide. The transmission at ribbon synapse relies on coupling of synaptic vesicle release and calcium influx in the control of exocytosis. The requirements for efficient synaptic transmission are high. First of all, synaptic transmission is highly dynamic and occurs at different time scales, both rapid and slow. Secondly, the efficient synaptic transmission implicates a controlled sequence of events, namely release, decay, and reconstitution of their macromolecular components. Thus, synapses continuously undergo structural changes, yet the juxtaposition of the presynaptic constituents and the postsynaptic receptor sites must be preserved. Thus, synaptic continuously undergoes structural changes, yet the juxtaposition. The functional contact at the synapse involves numerous different classes of molecules and their exact localization at the pre- and the postsynaptic region as well as within the synaptic cleft. Molecules participating in synaptic assembly that have been identified up to now are cytosolic, secreted, inter- and transmembranous, cytoskeletal, and extracellular matrix constituents. Some are common with others well-known cell structures, some are new and specific for certain types of synapses. Finally, we have to ask the questions: how is synaptic specificity achieved? Presently, research is directed towards the characterization of synapses of different neuronal subtypes using new molecular tools, markers for components, serving for their localization for genetic mutation, also in human. Moreover, and in particular, gene knockout experiments that result in specific changes in structure and function are of great value in the characterization of individual types of synapses. A preliminary list has been published last year by Furukawa. We are now arriving at the end of this introduction. I hope I was able to convince you of the impressive progression that has been made in understanding the structural and molecular preparative steps before neuronal transmission takes place. 2 points I would like to recall. First of all, correct intercytoplasmic arrangements, tethering of the synaptic components is a prerequisite for successful synaptic transmission. Second, precise contact and synaptic pairing of a particular presynaptic neuron with different postsynaptic neurons require specific molecular programs of the individual subtype. Certainly, we have learned a lot during the last decades, but there is still a long way to go. Thank you for your interest and have a nice day. Arrivederci.